So, hi everyone, I'm Kiarash Jamali, and I'm going to be talking on ModelAngelo, which is an automated atomic model building program for cryoEM, as well as protein identification. So, this is your cryoEM map, and you would like to build an atomic model. And for ModelAngelo, the way we do this is, in order to automate it, we split it up into a three-step process, where the first step consists of predicting residue positions. So these include both protein and now nucleotide residues. So we try to predict the centers of these residues, approximate location. And then the second step is this creates a graph, and we would like to optimize this graph and turn it into a full atomic model. Although what comes out of ModelAngelo is kind of like a probabilistic prediction. And this is the bulk of ModelAngelo, which is this graph neural network. And then the last step is then you take this probabilistic output and you use heuristics to actually collapse it down into an actual atomic model that with specified sequence and so on. Okay, so for the first step, where we want to predict the residue positions, we can parameterize this problem as a segmentation problem. Right? So you have your cryoEM map, which is a 3D voxel array, and you would like to basically answer the question for each voxel in your map, whether or not there exists a C alpha atom representing the center of your protein residues, phosphor atom representing the center of your nucleotide residues, or neither. And this is actually quite a difficult segmentation problem for machine learning, because for an average map where you have, if the voxel size is 1 angstrom and then you have a box size of 300, that's 27 million voxels, and then you would have maybe a few thousand residues. So it's a very imbalanced segmentation problem. And in order to solve this, we had to try with a lot of different loss functions. But at the end, we can train a segmentation model that takes a map and predicts both protein and nucleotide residues. And then this is where the second step begins. So you take the residue positions and you would like to actually build out the full atomic model from that. And in order to do this, this task requires the mixing of a lot of different kinds of information, right? So you have your cryoEM map, which is again a 3D voxel array. You have this graph that we just mentioned, which is this protein and nucleotide residue positions. And you also have your sequences, protein sequences available. So how you mix this information is the key to actually being able to get good results out. And the way we decided to do it is to have this graph neural network that has kind of 3 tracks where information can flow from each different type of input into these residues features. So each residue has a feature vector that represents what ModelAngelo knows about it. And at each step of this, one of the types of input information gets extracted from that and placed into the feature vector. And this keeps getting updated throughout the network until we can actually use it to predict things about our atomic model. So for example, the first module is the cryoEM module. And this is going to be taking the cryoEM map and trying to extract features from it that for each residue. And the way that works is with this algorithm called attention. And basically, for example, in this part, we take each residue and we sample, interpolate a cube of density centered at that residue and also aligned with the backbone at that residue. We put it through a convolutional neural network and we get a feature vector out of it. So this can give you information such as what the side chain looks like, what the backbone orientation looks like at that point, and so on. And then we also want to know information about whether residues connect to each other or not, because just from the output of the segmentation, all you have is a bunch of nodes in space and you don't know what connects to what or what the direction is and so on. So in order to get this kind of information, we look at the 20 nearest neighbors of each node, and we connect them with a line, and then we interpolate a rectangle of density between them, and we put that through a convolutional neural network, and we get another feature vector. And all this information gets extracted and mixed through these feature vectors, and then you update your node features, which in the beginning start off with zeros, but then with application of each one of these modules, there's more and more information coming in. So the second part is the sequence module. And, there's this ESM fold paper that's really interesting, where just from single sequence, single protein sequences, not the MSAs like AlphaFold, and with these protein language models, they're able to predict structure. So there's a lot of structural information just in these sequence representations. And so this is obviously also going to be useful for model building. So the way we use that is we put the sequences through these protein language models, we get a sequence embedding for it. And then again, these node features come in, we look through the sequence embedding to see what parts are most relevant for each node. And then we use that information to update the node feature again. So now you've mixed both cryoEM information and sequence information together. Lastly, we also want to know geometrical information about our model. And this happens through this spatial invariant point attention module that's very reminiscent of the one in AlphaFold. And so the way this works is you get your node features and you predict points in space, and then you look to see what residues are close to those points in space. And if those residues exist, you take the information from those residues and you bring it to the residue you're centering at. So what this is really doing is it's looking for repeatable kind of geometric information that exists in your model. And if that exists, it learns something about what the environment is. For example, in an alpha helix, there's very repeatable things going on, and that would give information to your node about where it should rotate and so on. So, and then that's how the spatial IPA works, is with distances against other residues. So this is the graph neural network. And so far, all I've said is how these node features are basically getting enriched after each application of each module. And then this also happens in 8 layers. So this goes back from the IPA all the way to the cryoEM again, and so on. But how this actually results in an atomic model is you take these features from the graph neural network and you put it through different modules that each update a specific thing in your initial model. So for example, the backbone frame module rotates the backbone frame triangle for each of these using the node features. And so you can think that like at each update, the model that was initialized kind of randomly slowly starts refining. And ModelAngelo also predicts a confidence measure for each residue, which is based on how much it predicts the model is going to be wrong compared to the deposited model. And this is also coming from the node features, as well as probably the most important, which is the amino acid probabilities for each residue, right? Because again, we don't know what residue types exist until ModelAngelo has predicted them. Okay, so I explained all of the different modules, and you might be wondering which one of these is actually important and which one isn't. And it turns out they're all quite important, especially as the local resolution gets worse. So here, B factor is a proxy for local resolution, and what we're judging is how complete the model is and how correct it is. And you see that as you remove different modules, especially at the lower resolutions, performance really suffers. At higher resolutions, you could probably still do an okay job just with the cryoEM module, but then at lower resolutions, it's obviously hopeless. Okay, so that was for the second step, but how do you actually get an atomic model out of these predictions, right? And that's where the post-processing comes in. And so we have a probability associated to each residue based on what residue type we expect it to be, but how do we actually know what it is specifically? And so you could just take the highest probability per prediction for each one of them. But this, you're losing information here because you are also given access to the sequence. So what you want to do is you want to look at the sequence and see what is the best match to the sequence you have for each chain and use that information to determine the sequence for each residue. And the way we do that is we take the amino acid probabilities that ModelAngelo predicts, and we use these as the emission probabilities of a hidden Markov model. And the transitions between the states are actually based on the confidence measures that we talked about before. So as the confidence gets worse, the probability of it being a match state goes lower. And so once you have your hidden Markov model, you can very easily do like a hidden Markov model alignment against the sequence file. And then using that, you can fix ModelAngelo's predictions. And also you can start pruning away parts that ModelAngelo has built, but don't seem to exist in the sequence file. So this is the full pipeline for ModelAngelo. And so now we are going to look to see how well it performs. So ModelAngelo's training cutoff date was April 2022. And then for structures released after this, we looked at structures that had resolutions better than 4 angstrom in the EMDB that also had a related PDB entry. And then we did a very stringent sequence similarity cutoff to what we had in training. So anything above 10%, we didn't include in our test set. And that gives us a test setter of around 170 entries. And so here we use the Q-score of the deposited model as a proxy for the local resolution. And as you can see for proteins, as the Q-score gets higher, what you'd expect is, ModelAngelo's built, you get higher completeness out of what ModelAngelo built, which is the case. And also the backbone RMSD for ModelAngelo's prediction also goes down. And then on the right, it's the distribution of residues that ModelAngelo builds versus ones that it misses. And so you can see that when the Q-score is higher, there's a lot more residues that ModelAngelo builds. But then as the Q-score gets lower, the ones we miss dominate. And this slide is comparing the refined models for ModelAngelo and the deposited model, and comparing both the Q-scores and the FSCs. And basically, when ModelAngelo builds something in these higher resolution maps, we do as good a job as the deposited model for proteins. So that's encouraging. And now some examples for the nucleotides. So the nucleotides, there's just not many maps that passed our stringent criteria for in the test set. There was only something like 2,000 residues. So instead, we did a study, like a small scale study on ribosomes. And here you can see one. So the green coloring is nucleotide that ModelAngelo has built. The blue is proteins that ModelAngelo has built in this ribosome. And then the black outline is the deposited model. And so you can see on the right is the deposited model that's been colored by the Q-score. So where the Q-score is low, so the local resolution is bad, ModelAngelo does not build it, but otherwise it seems to do an okay job. But this is like a very high resolution map. For this other structure, the resolution was worse. It's like 3 angstrom, and you can see it in the Q-score coloring. And even here for the backbone of the nucleotides, it seems to have done a good job, but you can see now that the sequence for the nucleotides is getting more challenging. And that's the main drawback with the nucleotides is that even when we have high backbone completeness, what we see something that we don't see in the proteins, which is that the accuracy of the sequence matching is not 100%, which it's potentially because there's just not much information when we want to do the HMM alignment against nucleotide sequences, because often all you can see from the map at these resolutions is whether something's a pyridine or a pyrimidine. And that just means that for each residue, there's one bit of information, and that's not enough to do a high kind of likelihood alignment against the sequence. But still, for the backbones, the completeness is high, and also the backbone RMSD for higher resolutions is low. So that's good. And also something really interesting that we also have is you can also do protein identification with ModelAngelo. So if you look at the graph neural network here, if we just retrain this without including the sequence module, then you don't need sequence as an input. Now, we know from the ablation study that the results are worse at lower resolutions, but if you don't have access to the sequence, you might be able to get the sequence from the map if the resolution is high enough, right? So the way we do that is we train ModelAngelo without the sequence module. We get the predicted amino acid probabilities. We build a hidden Markov model just as in the heuristics for the post-processing. But then instead of doing an alignment against the input sequence file, we do a search against, for example, a proteome of the organism you're interested in. And then we might be able to find the proteins that consist in that chain. So for example, this phycobilosome structure that recently came out from the Sui group in China, it's the biggest entry in the for cryoEM single particle right now, and it's more than 16 megadalton. It's a huge structure, and if you read the paper, it's really fascinating how many proteins they found. There's 80 unique protein chains in there, but 6 of them they weren't able to actually solve. They didn't know what the proteins were. So they did trace them by hand, but they couldn't figure out the sequence of the protein. And we were able to, using this strategy, with high confidence, determine all 6 of these proteins, and then resolve the full phycobilosome with the new sequences. And that's what you see here. So the green is what ModelAngelo built with the sequences, and the orange is what was hand traced. And for some of these, the density is quite poor. So it's really interesting that we were able to get the sequences with high confidence. And also you saw Alan earlier today show the ciliary axonemes. And these also have a few unidentified proteins still. And we were also able to do the same strategy and get these proteins that were later also, we can confirm these with mass spec data as well with Alan's help, and to make sure that these are correct. So these are the 2 radial spikes that he showed, and 4 new assigned proteins. And this is the central apparatus, so 2 new proteins here. And for some of these, actually, we were able to find the sequences with quite short ordered regions of the proteins. So that was cool. And so, yeah, for conclusion, we can say that when the resolution is high enough and the sequence is known, we can reach a human parity in building proteins with ModelAngelo. The nucleotide backbones are of good quality, but the sequence remains a challenge even in high resolution maps. And it can be argued that we're better than humans at identifying novel proteins from the map when the resolution is high with this strategy. And yeah, I encourage everyone to try it out, and please let us know what you think. It's open source software, and version 1.0 that includes the nucleotides and protein identification will be released very soon. And I'd like to thank my supervisor, Shores, co-author Dari, and our collaborators Lucas Shell, Alan Brown, and Rui Zhang. Thank you.